everyone, and welcome to Chapter 4, Part 1 of 2. We're going to look at genetics and cellular function. Now, as we know that DNA and genes is extremely important in understanding various diseases such as diabetes, color blindness, cystic fibrosis. Now, if you guys do not know or do not remember, there is a gentleman by the name, a scientist, an Austrian monk by the name of Gregory Mendel, who played a major role in us understanding chromosomes and the relationship between chromosomes and behavior, as well as any traits being passed on from parent to the child. And he's so famous for his experiments on the garden peas. Now, when we look at Mendel's genetics, it deals with parent-offspring and larger family relationships to discern and predict patterns of inheritance within a family line. Now, cytogenetics uses the techniques of cytology and microscopy to study the chromosomes and their relationships to hereditary traits. Molecular genetics, it uses the techniques of biochemistry to study the structure and function of DNA. Then we have genomic science or genomic medicine. This comprehensively studies the entire DNA endowment of an individual, and then how it influences health and diseases, and how its knowledge can be used to prevent, cure, or even treat various diseases. So these are the subdivisions that we have from just dealing with genetics. Now let me look a little further. We're a number of scientists, but this is a Swiss scientist or a Swiss biochemist who studied the nuclei of white blood cells extracted from pus and used hospital bandages, and later the nuclei of salmon sperm. Now, since both cell types offered large nuclei with minimal amounts of contaminating cytoplasm, both of them were utilized. But then in 1869, he discovered an acidic, phosphorus-rich substance he named as nuclein. Now, he correctly believed that this to be the cell's hereditary matter, but of course he had some individuals, some scientists that did not believe him. But then we now know that what he discovered was correct. So by the 1900s, we have biochemists are now using the basic components of our DNA, which is sugar, the phosphate bases, and the nitrogenous bases. And in 1953, we had the overall structure of the DNA that was learned. Alright, so let us look at the DNA structure as a whole. Basically, what does it look like? What is it composed of? Well, it's a long thread-like molecule with uniform diameter, but it varies in length. Okay, so we have about 46 DNA molecules in the nucleus of most human cells. The total length is approximately 2 meters, and the average DNA molecule is 2 inches in length. So, what does it consist of? Well, we see that DNA and other nucleic acids are composed of nucleotides, each of which is composed of a sugar, a phosphate group, and a nitrogenous base. Now, when we look at the nitrogenous bases, we have cytosine. Over here, we have cytosine, thymine, uracil, and these are pyridamines. Then we have our adenine or adenine, however you like to pronounce it, adenine and guanine. And these are considered to be your purines. So basically we have two types of nitrogenous bases. first type is our purines, so they're adenine, guanine, and our second type is our pyridamines, which are the cytosine, the thymine, and the uracil. Yes, they do have a single ring. The purines do have a double ring. But when we look at the bonds that are between them, the hydrogen bonds that are between the adenine and the adenine and the thymine, because these are only two that will match up together, that will pair, are two hydrogen bonds. And with the guanine and the cytosine, which will bond, they have three hydrogen bonds. So let's see next slide here. All right. Here we have our adenine and our thymine. Yes, they are in they're different types. The adenine is with your purine, your thymine is with your pyridamine, but when it comes to when it comes to the DNA structure and then pairing, you have one purine and one pyridamine that will bond together to form this staircase of the DNA. So when they bond, the adenine and the thymine, they have a double hydrogen bond, which is signified here with these particular lines. So this is an example of one hydrogen bond, and there's your second hydrogen bond. Now when it comes to your cytosine and your guanine, when they pair, you have three hydrogen bonds, which are going to keep them together. Okay. Now the adenine, thymine, cytosine, and guanine combinations, they are called base pairs. Now when we look at the structure of the DNA, yes, we have our steps. It does look like a staircase. So we have our steps, and then we have our backbone. And the backbone, it composes of a phosphate group alternating with a sugar deoxyribose. So here's your phosphates, and here are your sugar groups, which would be your deoxyribose within the DNA. We're just doing the DNA, okay? Alright, so I talked a little bit about your base pairing. I kind of jumped ahead. I talked about your base pairing. You already know that when the guanine and the guanine and cytosine pair, they're held together by three hydrogen bonds. When the thymine and the adenine pair, they're bond together by two hydrogen bonds. Okay, so we have a law of complementary base pairing, and that allows for the prediction of the sequence of one strand if the other strand sequence is known. So if you know that one strand is guanine, thymine, guanine, adenine, it's normal, or it's natural to know that, hey, the second strand will be cytosine paired with our guanine, our adenine paired with our thymine, our cytosine paired with our guanine, and our thymine with our adenine. So let me see if I can pull. Okay, let me get a color here. Let's get a blue. So if I give you one strand of DNA and I say adenine, thymine, guanine, cytosine, adenine, guanine, what will be the second strand? The second strand, of course, when we deal with our pairing to complete this DNA structure, will be what? Thymine. Oh, let's change colors. Okay, this will be our thymine. This will be adenine, cytosine, guanine, thymine, and our cytosine. So that is the rule when it comes to the law of complementary base pairing. You're able to determine what the entire what the two strands will be, what the second strand will be, if you have the proper coding for the first strand. All right, so let's look at a few terms. We have genes. What's genes? Well, genes is genetic information or genetic instructions for synthesis of proteins. When we look at gene, it's the segment of DNA that codes for a specific protein. Then we have a genome, and a genome is basically dealing with all the genes of one person. Now, there's approximately 20,000 to 25,000 genes that an individual will have. 98% of it is non-coded DNA, so their role is basically to help with the structure of the chromosome, to regulate the gene activity. Then, of course, you have the junk DNA. And then the last 2% is the total DNA. Now, when we look at the discovery of the double helix, our DNA, there are three individuals that come to play. It's Watson, Crick, and Wilkins. Now, yes, four people, and of course, Franklin, Rosalind Franklin. Unfortunately, she was not awarded the Nobel Peace Prize because they do not award it to individuals that have passed away. But the story starts off with individuals in London. You have Watson and Crick, who worked together at the Cambridge University, and they were doing their research in DNA structure. They're trying to get a better understanding. Now, this is back in 1951, where things are slowly but surely progressing to what we know now. Now, we had another individual, Rosalind Franklin, who was also working in London, but she was at the King's College in London, and she was using a technique called the X-ray diffraction, and that was helping her to determine the structure of the DNA, how it repeats itself from especially when it comes to the backbone of the DNA, where you have the sugar, the phosphate, sugar, phosphate, sugar, phosphate, how everything is just alternating. So she had her picture and getting a better understanding of it. And at the other, Cambridge University, you had Watson and Crick doing their experiment. Now, unfortunately, Wilkins, he, who was working with Rosalind, he stole, can't say borrowed, the book says he took it without permission, but if you take something without permission, it's stealing. So he stole the picture that she developed and took it over to Watson and Crick at the other university. And when Mr. Franklin observed, Dr. Franklin, observed the X-rays, not Dr. Franklin, I'm sorry, when Dr. Watson observed the X-rays that Wilkinson showed him, he realized, hey, this is the missing piece to my puzzle. They hurried up, they got the information together, and decided to, hey, let's go post it, let's go print a paper. This is back in 1953 now, showing that, yes, we have discovered it all, and they gave no credit to Mrs. Franklin, Rosalind Franklin. It's pitiful that your own colleague, or your own colleague working with you, would actually betray you and take your information to another college and try and get credit for it. Not try and get credit, but they got credit for it, which is horrible. So unfortunately, not unfortunately, but Rosalind, she found out about it, and she went ahead and she rushed her paper off. So she was able to get her paper off around the same time that these three men were able to to establish and get their paper out. Now, unfortunately, Mrs. I mean, not Mrs. I keep saying Mrs. Dr. Franklin, she was not honored, like I mentioned earlier, and she died at the age of 37. And they're saying it had to be due to utilizing this this X-ray machine, the X-ray diffraction diffractory machine that she was utilizing to get these pictures. She died of cancer. Okay, so make sure you guys know who these three individuals are. You will see it again. Alright, so let's dive into the DNA structure itself. We have something called the chromatin. What is the chromatin? You guys have read ahead, but DNA is complex with proteins to form a fine filamentous material called chromatin. Now, human chromatin consists of 46 long filaments called chromosomes. Alright, let's see again. So here's my chromatids. The 46 of these that make up my chromosomes. Now, the DNA is about 2 meters in length. If fully unwound, may be packed into the nucleus. Now, viewed with electron microscopy, the chromatin has an appearance like beads on a string and is divided into segments called the nucleosomes. This is the structure from here to here. Now, each nucleosome consists of a core particle, like this core particle, and a short segment of a linker DNA. Now, the core particle is a disc-shaped cluster of 8 proteins called histones, with the DNA wound around it like a ribbon around a spool. Now, the nucleosomes are arranged in like a zigzag pattern, folding the chromatin like an accordion. The folded strand is then packed into irregular loops and coils that make up the chromosome. Each chromosome is packed into its own region called the chromosome territory, permeated with channels. Now, when a cell is preparing to divide, it copies its entire nuclear DNA. Each chromosome is made up of 2 sister chromatins. Then they coil even further. Now, these chromatins are visible in the light microscope. Now, in early cell division, a chromosome is compacted to its greatest extent. Each chromatids are joined at a centromere. So here's our sister chromatids. Here is this area here is considered to be our centromere. Okay. Now we have our kinetochore, and those are protein plaques on either side of the chromatin of the centromere. Sorry. Now let's look at our RNA. What is an RNA? Now, when we look at RNA, we know that it is, hopefully you should know, it's termed for ribonucleic acid, and it's produced in 3 types. We have our messenger RNA, which is termed abbreviated mRNA. We have our ribosomal RNA, or RNA, and then we have our tRNA, which is our transfer RNA. The RNA and the DNA have significant differences, although both are nucleotides. Are nucleic acids. Now, an RNA is much smaller than the DNA, ranging from 70 to 90 bases in the tRNA to over 10,000 bases in a large mRNA. The DNA averages more than 100 million base pairs. So we're looking at quantity here. Now we're going to look at what it looks at, your structure. The RNA is a single nucleotide chain, not a double helix. Third difference, the sugar in RNA is ribose, not the deoxyribose, which you'll find in D and A. Now, then we have the thymine. Now, we talked about the adenine, thymine, guanine, cytosine. Now, in RNA, the thymine is replaced with a U, which is your uracil. So, I can grab. It's not showing up. Okay. Oh, here we go. Alright, so with our DNA, we know we have our thymine, adenine, guanine, and our cytosine. And we know that they're going to pair accordingly. Sorry for my writing. With adenine. Adenine pairs with our thymine. Guanine pairs with cytosine. Cytosine pairs with guanine. But when it comes to our RNA, what do we have? We have, I'll use the same one here. Instead of using thymine, we have uracil. We have adenine, guanine, and cytosine. There's no thymine in our RNA. Always remember that. So how would it pair if we're matching it up, or as we find out later, forming a second strand? How would it pair? So since U replaced T, U will pair with A will pair with U, G with C, and C, our cytosine, with our G. All you're doing is just replacing the U, replacing your T's over here with our U's, with our uracil. So our thymines with our uracils, okay? So I'm going to switch to the other page. Okay, so in your textbook you should have a chart or a table on page 117, table 4.1, and it will describe to you or give you the comparisons of our DNA and our RNA. Looking at the sugar, the type of sugar that's used within the strand, the type of bases that it has, the number of nitrogenous bases, the number of nucleotide chains, the site of action for each one take place. So we see for DNA, it functions in the nucleus and it cannot leave. The RNA is going to leave the nucleus, which you'll find out later on, and it functions mainly in the cytoplasm. The function for DNA codes with synthesis of the RNA and the protein, and then the RNA carries out the instruction of the RNA, and then it starts to assemble proteins. So this stuff here will actually make sense when we talk about translation and transcription a little bit later. Alright, right, so let's look at a gene. What is a gene? A gene, previous definition that we have, is a segment of DNA that carries the code of a particular protein. Now, the classical concept of a gene was an abstract unit of hereditary by which a trait passes to offspring. We now define a gene as an information containing segment of DNA that codes for the production of a molecule of RNA, which in most cases does not play a role in protein synthesis. Now, the 46 chromosome comes in two sets of 23. It comes in two sets of 23 each, one set for each parent. All the DNA in one 23 chromosome set is called the genome. Now, the human genome consists of about 3.1 billion nucleotide pairs. The Human Genome Project revealed the base sequence of more than 99% of the genome. Genomics is a new field of biology that studies a genome and how its genes and non-coding DNA interact. Now, revelations from this Human Genome Project included 6 major findings. What are those findings? Humans have fewer genomes than the 100,000 previously thought of. These genes generate millions of different proteins. A single gene can code for many proteins through alternative splicing of the mRNA. Third, genes average about 3,000 bases long, but range up to 2.4 million bases. Next finding, all humans are at least 99.99% genetically identical. But even this small variation means there is a probability to differ from one another in more than 3 million base pairs. Contributions of these differences are called single nucleotide polymorphism. Just right here, single nucleotide polymorphism, and it accounts for all human genetic variations. Next finding, our fifth finding, some chromosomes are gene rich and some are poor rich. Sorry, not poor rich, are gene poor, I'm sorry. The sixth one, we now know that there are more than 1,400 disease-producing mutations, which opens the door to a new branch of medical diagnosis called genomic medicine. Now, before this project, we knew locations of fewer than 100 disease-producing mutations. So this project has opened the eyes of many scientists, and of course the biology scientific field. Alright, so let's look at genetic code. The genetic code is a system that enables 4 nucleotides to code for the amino-base sequences of all proteins. The minimum code to symbolize 20 amino acids is 3 nucleotides per amino base. Now, each group of 3 nucleotides coding for amino acid is called a base triplet. Okay. Now, the messenger mRNA contains a mirror image of each base triplet, and this group of 3 bases is called a codon. The possible combination of 3 bases would yield a maximum of 64 codons. Therefore, some amino acids are specified by more than 1 codon, which you will see in Table 4.2 on page 119 in your textbook. The code also contains stop codons that end the translation to amino acids, and a start codon that also codes for methionine. I keep pronouncing this wrong. Methionine. Okay, so in your textbook, I'm not sure if I have it here in the book, my slides. I don't. But in your textbook on page 119, you'll see the table 4.1, and it'll give you the various codes for starting. You already know that methionine, which is abbreviated MET, is your start codon. Now, the base triplet for that is RT. Is our TAC. So whenever you see your TAC, you know that, hey, this is our methionine. And this is what's going to start the particular sequence. Now, I talked about earlier about 3 nucleotide codings. Each group has a 3 nucleotide. Now, here's your nucleotide. We have 1, 2, 3. So this is group 1. Oops. Then we may have another group, which would be CCT. That's our second group. Our third group could be CCC. Fourth group could be T, G, and G. So for this particular chain that we're trying to create, we just have 4 particular codons or base triplets. We have base triplet here, base triplet here, here, and here. So we have 4 types of base triplets. Now, when we mirror it, what will we have? We will have a different color. When it goes to the system, we have A. Sorry. We'll have A, T, and G. This one will be G, G, A, G, 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 then A, C, and C. So this is the mirror that I was referring to. And this will give us the codon. So this will be our base triplets, what we're starting off with. And when we're trying to get this genetic code, it mirrors itself, giving us our codon for our mRNA. So this is the base triplets for our DNA. Now we need to get the base triplets for our... Oops, that's not A. Sorry, this should have been U. This should be U. Now this should have been A, U, G. I'm sorry. This should have been A, and this should have been U, G, because there's no thymines in our mRNA. So we have our base triplet for our DNA, and we're trying to make our mRNA, which is our messenger mRNA. And when it mirrors itself, we have various codes. So with our TAC from our DNA, we are going to have AUG, Our CCT, we will have GGU. There should have been a U, I'm sorry. Oh, that's right, I'm sorry. GGA. Then our CCC from our DNA is GGG. And then from our TGG will give us ACC. So this will be our codon of our mRNA. And each of these have different names. Let's scratch this off here. Our AUG is our methionine. Our GGA is our glycine, which you see in your textbook in that table 4.2. Our CCC, which changed into GGG, is also our glycine. And our TGG, which changed to ACC, is now our threonine, abbreviated as THR. And this will be your amino acid. So, from the base triplet and it mirroring itself, the base triplet of a DNA, and it mirroring itself, forming the codon of our mRNA, we were able to get our amino acids. These are the chains that we have producing. Forward a little bit too much. Alright, so DNA and RNA collaborate to make these proteins that I was telling you about. These amino acids, we already know that they are proteins. They eventually make these proteins. Now, the genetic code specifies which proteins a cell can make. Now, different cells have different active genes. A given cell uses only 1/3 or 2/3 of its genes. Now, when a gene is activated, an mRNA copy is made. So when those genes that I just showed you, the DNA, the base triplet of the DNA, when it's activated, that's when we're going to get this mRNA copy. And this is what migrates to the cytoplasm, where it's coded by a ribosome. So we have the activated gene, we have our messenger mRNA is what helps it mirroring the gene, and it migrates from the nucleus to the cytoplasm, and its code is read by the ribosomes. Now we have the ribosomal RNA. We have our ribosomes, the cytoplasmic granules. They're composed of the rRNA and enzymes. Our transfer RNA, they're going to deliver these amino acids to the ribosomes. Now the ribosomes, they're going to assemble amino acids in order directed by the codes of the RNA. So like in the last slide or so I was showing you, where you had the AUG, which is our start, our AUG, our I think it was to say GGA, then R to say GGU, and I'll do one more, our CA. These will basically code for a particular amino acid. So our AUG is our MET. I'm using abbreviations right now. Our GG, that is a G. Our GGA is our glycine. I'm using abbreviation. Our GGU is also glycine. And then our GCA is our ALA. These are all abbreviations. So when all these are coded and we have our particular amino acids all lined up, they're going to align in order. You're not going to have our ALA starting the code. No, you're going to have MET, our glycine, and then our ALA. It won't switch around. Whatever is coded first, it will go in that particular order. Whatever is coded by that mRNA. So when we look at the process of protein synthesis, you have your DNA. It changes and it converts, or it's coded, mirrors itself to mRNA, and the mRNA helps with producing the proteins needed. Now, it goes through various steps. We have our transcription and our translation. From DNA to mRNA is transcription. And this is what occurs in the nucleus. The second step, our translation, this is the step from mRNA to our protein. And this is going to occur in the cytoplasm. Now, 10% to 15% of proteins are synthesized in the nucleus. The remaining is in the cytoplasm. Now, if we have any DNA that's too large to leave the nucleus and participate, we do have DNA, that is too large to leave the nucleus and participate directly in the cytoplasmic protein synthesis. It is very necessary to make a small mRNA copy that can migrate through this nuclear pore into the cytoplasm. Now, transcription is that very first step, correct, that we talked about. And this is copying genetic information or genetic instructions from our DNA to our RNA. Now, what happens here? We have a structure called the RNA polymerase. Now, this is an enzyme that's going to bind to the DNA, and then it's going to assemble the mRNA. So, certain DNA sequences such as TA, it instructs the RNA polymerase where to begin. Just hold on 1 second. Okay. I actually had to find a picture for you guys and see if I can. I had to just input it, so I have to repost the video. I mean, it's not a video. I have to repost the PowerPoints. I thought this picture wasn't there, but it's not. But this picture says 1,000 words, speaks 1,000 words. So let's deal with transcription. Already know what transcription occurs within the nucleus and translation occurs within the cytoplasm, correct? Okay. So here's the nucleus, cytoplasm out here. Here's our DNA strand. Here's our RNA polymerase. Here's our RNA transcript, which I'll get into details, and then we have things called axons and axons, axons and exons. So, axons and introns, I'm sorry. Alright, so let's start off. When we talk about transcription, I already said where does it occur? In the nucleus. And we're dealing with the process by which DNA is copied into RNA. Now, our goal is to make proteins. When we make these proteins, it has to start somewhere. And this is the transcription stage, stage 1. Now we have our RNA polymerase, which is an enzyme that's going to bind to DNA. That's going to bind to the DNA and start assembling this RNA strand. Now, certain DNA sequences, such as our TA, they're going to instruct this RNA polymerase where to begin. So on the strands, remember we talked about adenine, thymine. Now, if certain ones, if it sees TA, it knows, hey, guess what? We need to start at this point. Not at GC, GA, GC, GT, but we're going to start at TA. So once it sees or starts to read that particular code on the DNA, the RNA polymerase will attach itself to the DNA strand, it will unwind, it needs to unwind first that small little section, and it will bind to the DNA, unwind it, then it will start coding for what it needs to code for. Remember, we were talking about mirroring. Mirroring. Now this enzyme, it opens up the DNA helix, like I just explained, and it starts to read the base in one strand, and then it matches them with the complementary bases. So if we have a TAC, for example, TAC for our DNA strand, what will be the RNA strand? The first code will have A, U, G. Remember, there's no thymine, and the thymine is replaced with uracil when we deal with the RNA strand. Okay, so it'll code for that particular strand, giving us a complementary base. Now, at the end of the gene is a terminator sequence that signals the enzyme to stop. Now, it all depends on what the code is. So you're not going to have a 30, 50 foot RNA. RNA transcript. Some of them will be 3 inches, some of them may be 50, some of them may be, just say, maybe 20 inches. It all depends on when that stop codon or the stop sequence lines up on our DNA. Okay? Now, the RNA produced is a pre-mRNA. That's what it's called, a pre-RNA. That contains sense portions called axons and a nonsense portions called the introns. So here's our introns, and here are axons. Okay? Now, enzymes excite the introns, and they're going to splice together the axons. Now, through alternative splicing, a gene can code for more than one protein. So it's going to splice it here, called alternative splicing. And then they're going to splice together the exons. It's going to splice it here, and these axon portions will combine together to form what will be leaving the cytoplasm. Now, when this is formed, we have translation. Translation is going to occur because this particular pre-RNA transcript is ready to leave, and is ready to leave, and is in the process of leaving the nucleus, this small pore, into the cytoplasm. It is our mRNA now. Now, translation is a process by which the language of nucleotides is converted into the language of amino acids. Now, 3 components are needed for this process. What do we need? We need our mRNA, which begins with a protein cap, which is right here, where a ribosome will begin translation. Okay. The second component is our tRNA. Here are little tRNAs right here. And what do we say those were? Transfer ribosomes. And it's a molecule that coils on itself to form an angular L shape that has a 3 nucleotide codon. So here's our little codons. Here's the L shape, and here are 3 codons. So that's just 3 codons on one end, 3 nucleotide anticodon, a 3 nucleotide anticodon on one end, and then a binding site for one amino acid on the other end. And the third component for this process is our ribosome. Ribosome, which consists of two subunits. It has a large unit, which is the larger part here, and a small subunit. And they're composed of RNA and several enzymes. Now the two subunits come together. So here's your large subunit, and here's your small subunit. Now when they come together, right here, they're going to form 3 pockets. We have 1, 2. You have site A, site P, and then you have site E, where the tRNA binds. Now translation is going to occur in 3 particular steps. First step is called initiation. Initiation starts when the mRNA passes through this nuclear pore. The small subunit binds to a leader sequence near the cap. The subunit slides along the mRNA until it reaches the start codon AUG, which you see here. So you notice this is the beginning part, this is the cap, but the ribosomes are not going to assemble itself on this mRNA until it reaches that AUG start codon. So you see how much of the code, or how much of this mRNA has passed through, has gone through? Alright, so what did it have? Oh, here's our AUG. So this is the start point. So the initiator tRNA now is just sitting in here, amino acid activator. This initiator tRNA with the anticodon AUG pairs with the very first codon. It positions itself into the P site of the joining large subunit. The assembled ribosome is going to slide along the mRNA and reads the codes. That is the initiation stage or step. The second step in translation is elongation, and this represents sequential ribosomal activity that increases the number of amino acids in the polypeptide chain. The next tRNA arrives carrying another amino acid. It binds to the A site. It's going to bind to that A site of the ribosome, and its anticodon pairs with a second codon. Now an enzyme in the ribosome transfers the MET, which is the very first one, the start codon, of the initiator tRNA to the amino acid of the second tRNA, and it creates a peptide bond, giving it a bipeptide. Alright, so the ribosome is going to slide down to read the next codon, which shifts the initiator tRNA without the amino acid to the E site, where it's going to leave the ribosomes. The second tRNA carrying the bipeptide, which is this one here, and the bipeptide, shifts from the P site shifts up, no, right here, shifts into the P site, opening the A site for a third ribosome or third tRNA site or tRNA to bind. So everything is shifting from one site to the next, from the A site to the P site to the E site. The A site is basically where it starts to initiate. The P site is when the amino acids are binding with each other, creating that bipeptide bond, or given a peptide bond. And when it has released its amino acid, it moves to side E when it's released. Then it is released. Now that bipeptide from this second RNA, tRNA, is now transferred to the amino acid of the third. And a new peptide bond is formed, creating a tripeptide. So that means you'll have 3. So all we're doing here is just forming peptide bonds and amino acids from the coding of our nucleotides. This is all translation. Now the process is repeated by the sliding of this ribosome. This ribosome is going to keep sliding to the next codon. Now the empty tRNA exiting, which is this one here, exiting this E site, it goes off to pick up another amino acid. Now some mistakes are tolerated at the third base of the codon, and this is called a wobble, and it allows I think it's 48 different tRNAs to pair up with 61 codons that represent our amino acids. The last stage, our third stage, is termination. It can't keep going on and on. It has to stop sometime. So termination is the process where the ribosomes reach a stop codon. The A site binds a protein called release factor instead of the tRNA. So you're not going to have this tRNA binding to this A site. You're going to have a release factor. Now this causes the release of the protein from the ribosome. The ribosome is going to do what? Just dissociate into the two parts, which will be the small and the large, that can reassemble at the same mRNA and start the process all again. If the protein is to be packed into a lysosome or created from the cell, the ribosome it docks on a rough endoplasmic reticulum, and the new protein kind of spools off into the cisternae of the ER instead of the cytosol. So yes, I am mentioning different parts of the cells, so you guys remember what these parts are. Now, one mRNA can hold 10 to 20 ribosomes in a cluster, and that's called a polyribosome. And an mRNA is translated by all of these ribosomes at once. So transcription and translation are sequential processes that begin with DNA and then end with a protein product. Okay, I believe I have some videos on this, so please look at it over and over and over again until you understand exactly what is occurring, because you will definitely see this particular process again, these two processes, transcription and translation. Okay, so that's the diagram. Here are the information. Here's the information for each of the steps that I just went over. Okay, so I'm not going to go through those. Various steps, elongation, translation. I had some diagrams. All right, here's some of the diagrams here, which your textbook would have. I had the other one, which I liked a little bit better. But you see the small little pores. that the mRNA would leave. This is of the nucleus. So you guys remember nuclear pores? You have the nucleus, the nucleolus. Okay, so these are the pores where you have the mRNA coming out of. So you can start this translation. Start in the translation process, okay? All right. 3. Here's your rough endoplasmic reticulum. Hope you guys remember the difference between rough endoplasmic reticulum and smooth endoplasmic reticulum. But you see that it does, once the proteins go through this whole process, forming these vesicles, and is released by the rough endoplasmic reticulum, forming our transport vessels so that it can be transported to where it needs to go. Okay. It's very important that you guys understand what is going on. Alright, so review of our peptide formation. What do we have? We start with our double helix, correct? Good, we start with our double helix, which is within the nucleus of cell. The second one is our 7 base triplets on the template strand of a DNA. So we just have 7 of them. We know that TAC is our signal for our polymerase to come along and start reading. Is our start. So once it reads TAC on a DNA, it knows, hey, okay, this is where we need to start, and it starts coding, and this would be the very first codon, AUG. Then it keeps coding, matching up. T with G with C with G, C with G, T and A, T and A, and it keeps matching, coding up. We know that A is not going to be matching up with T in our RNA. It's going to be matching up with the uracil. And it keeps coding on, okay? So the corresponding codons of the mRNA transcribed from the DNA triplets that we have here, okay? So this is our mRNA now. So number 4, the anticodons of the tRNA that bind to the mRNA codons. So for each codon that we have, for each codon that we have, we're going to have different amino acids. The UAC, here we go. It's going to carry them. And these are the same codons. This is from the same codons that were used, or base triplets that we used from the DNA to the mRNA. These are the same ones transferred that we're going to have these particular amino acids forming. Okay? So the amino acids carried out by these 6 tRNA molecules. The last step is the amino acids linked into a peptide chain. So these are the amino acids, the amino acids that are linked together that will pass through the rough endoplasmic reticulum. So this is just a review of a summarized portion of what happens from DNA, of transcription to translation. Okay. Alright, so we talk about protein processing and secretion. Protein synthesis is not finished. No, it is not. When the amino acid sequence or the primary structure has been assembled to be functional, it must coil or fold into precise secondary and tertiary structures. Now, in some cases, association with other protein chains, or it will start binding with other molecules. Alright, so we have these things called chaperone proteins. Now, these chaperone proteins, they guide a new protein strain into its proper configuration. Now, some of these are called stress proteins or heat shock proteins because they are produced under those particular conditions. Okay. The ER, the endoplasmic reticulum, and the Golgi complex or apparatus, they have important roles in protein processing and secretion. Now, a protein assembled on the ER, or I'm going to say ER. We're dealing with rough endoplasmic reticulum. Those proteins that are assembled on those, they the surface. Those proteins assembled on the ER surface. It moves into the ER cisterna, where it is modified by enzymes. This is called post-translation modification. So the ER is going to pinch off. ER is going to pinch off a bubble-like transport molecule coated with this clathrin that holds completed completed proteins. Now these are going to transport the vesicles. These transport vesicles may fuse and move to the Golgi apparatus. So here it is pinching off, and they're going to move to the Golgi apparatus. The cluster of vesicles fuse with the Golgi apparatus, which you see occurring here, and it's going to release its contents into the Golgi cisterna. The Golgi complex modifies proteins further, often by adding carbohydrate chains. Now, the Golgi apparatus, or the Golgi cisterna, furthest away from the ER, buds off and breaks off into the Golgi vesicles, which we see here. Now, some of the Golgi vesicles become lysosomes, while others become secretory vesicles that are going to merge with the plasma membrane, which you see here. The plasma membrane merge with the membrane, then it's going to release the cell products by exocytosis. So some become lysosomes and some become secretory vesicles. Now, proteins synthesized in this way have a variety of destinations, and they have a variety of functions. And you should be able to find that. Let's see, this table. Yep, on table 4.3 on page 125. You see the functions, destination and functions of the various proteins that are going to be released from this Golgi apparatus, from the secretory vesicles. Okay. All right. So the diagram here is basically explained in these last few slides. Alright, so let's look at gene regulation. I believe this is the last section for this particular lecture. Look at genes. They're regulated by a number of factors that turn them on or off as their products are needed. The production of casein by mammary glands cells, they produce an example of gene regulation. Now, the hormone prolactin that binds to receptors in the plasma membrane by a mammary gland cell. Now, the binding to receptor, they're going to trigger activation of the regulatory protein in the cytoplasm. The regulatory protein moves into the nucleus and binds to the DNA near the gene for casein and for casein, which is a milk protein. Now, this binding enables the RNA polymerase to transcribe the gene producing this mRNA. Now the casein mRNA, it moves to the cytoplasm and is translated by ribosomes on the rough endoplasmic reticulum. The Golgi apparatus, or the Golgi complex, it packages casein into these secretory vesicles. Now these vesicles are going to release this casein outside the cell, and it becomes part of breast milk. So here is the example of what is going on here. We have the hormone prolactin, which binds to the receptors, which is on the mammary gland, the plasma membrane of the mammary gland. Now the binding of the receptors, what is it going to do? It's going to start triggering the regulation of the protein in the cytoplasm. Of course, you need ATP. ATP is always needed. And with that ATP being utilized, you have ADP giving us ADP plus our phosphate group, and this phosphate is utilized to help with regulation of the protein, which is our transcription activator. Now the regulatory protein, it moves into the nucleus, and then it binds to the DNA near the gene for casein, which is that milk protein. Now this binding, what does it do? It's going to enable the RNA polymerase, which is right here, this green object in this diagram, to transcribe the gene, producing our mRNA for the casein. This casein mRNA, it moves into the cytoplasm, passes out through the pores, and is translated by ribosomes on the rough endoplasmic reticulum. So slightly different from the other one that we talked about, from protein, from DNA translation and transcription, slightly different. Now the Golgi apparatus, or the Golgi complex, is going to package these caseins into these secretory vesicles, and these vesicles are going to release the casein outside of the cell. Here, outside the cell, and it becomes part of breast milk, and releases it through the process of exocytosis. Alright, so regulatory proteins can turn gene transcription on or off in a number of ways, including attracting, and we go to back. So it's attracting and positioning the RNA polymerase, and coiling or uncoiling DNA in a nucleosome to make it accessible. Now compounds other than proteins are synthesized in cells and are made through the activity of enzymes, which are proteins encoded by genes. Now there are no genes for stored testosterone, but a cell of this testes I'm sorry. Basically going to convert the cholesterol into testosterone if the genes for the enzymes are activated. Now the DNA codes for RNA and protein synthesis does it indirectly. Sorry, the DNA codes for RNA and protein synthesis, but it indirectly controls the synthesis of a wide variety of substances. Yes, there's a lot of information. There's a couple of processes or steps that need to be take into consideration when you're studying. Please study the details of translation and transcription. We will see again details. Make sure you know the difference between RNA and DNA when it comes to structure and function and location. Please look at all of that information. If you have any questions, do not forget to post and look at part 2 of the video. Thank you very much.